The elusive magic bullet, a delivery system for medicine that only targets the bad cells. A system as accurate as an arrow and as precise as Tiger Woods. A system to make the treatment of life-threatening diseases more effective than ever before. The magic bullet sounds like the perfect choice for treating diseases. You might even call it the Holy Grail of medicine. The idea was first initiated by Paul Ehrlich over 100 years ago.
Professor Ehrlich famously transformed the ancient art of finding drugs in plants by the science of producing drugs from chemical dyes. In doing so, he changed the dye industry into the pharmaceutical industry. He discovered that a chemical dye could selectively bind to its target, something which was later called a receptor. He then reasoned that drugs could act as a magic bullet, only hitting the target receptor without causing any side effects. Now, more than a century later, we still have not managed to realize this magic bullet. When we look at genetic diseases, for example, for many of those we know exactly what causes the disease, and we also have the tools to correct such mutations. Yet, we fail to translate that into an effective therapy. So far, only one treatment has been approved in Europe. The difficulty often lies in finding the right delivery system that can safely and effectively deliver the therapeutic genes into the right subcellular compartment of the affected cells.
Every day we ingest several grams of foreign DNA and RNA from the food that we eat. This never changes the DNA in our cells because our body is designed to destruct all exogenous DNA in order to prevent diseases. It's clear then that if we want gene therapy to work, we have to beat our own defense system. A way to overcome some of the obstacles is to inject DNA molecules right into the bloodstream. But this is not enough. First, after injection, the foreign DNA is rapidly degraded by enzymes in our blood. For those DNA molecules that survive enzymatic attack, there are several more barriers at the cellular level. The DNA molecules are too large to penetrate cell membranes, and as such cannot gain access into the cell's interior.
Efficient delivery systems for DNA and RNA can be found in nature in the form of viruses, bacteria, and membrane vesicles. In fact, more than 8% of the human genetic makeup comes from remnants of viral infection. This shows us that in principle it is possible to deliver exogenous DNA. Indeed, many researchers are already making use of viral vectors. My research is an attempt to copy these biological systems in a synthetic way, an example of so-called biomimicry. Packing the gene in an artificial nanosystem leads to less immune responses and ultimately to better results.
To make such an artificial delivery system, first, a vesicle is created around the gene. Then we add a protein, making it able to home to the target cells. Once the cell binds to the target receptor, the vesicle is eaten up and ends in a bag known as the endosome. Within the endosome, the vesicle surface changes, thereby enabling the disruption of the endosome and release of the vesicle contents into the cytosol. After the contents are released, an exposed peptide flag recruits endogenous proteins to transport the DNA into the nucleus via the nuclear pores, small gated channels in the nuclear envelope that are normally closed for large DNA molecules. Once the DNA is in the nucleus, it can be read, leading to the formation of proteins via an RNA intermediate.
This technique works perfectly well on cells in culture. However, in the human body, there are still some challenges to overcome.
We all want this magic bullet to be realized. We want it to become as accurate as an arrow and as precise as Tiger Woods. Bright minds from various backgrounds are needed in order to transform gene therapy from a headline into a leading method of treatment.